Shut up and sit down.
Welcome to Transposable Element Detection Methods via the McClintock Pipeline. I hope you enjoy. I'd like to begin with some information about the structure and function of transposable elements, which I will also refer to as transposons. Transposons are mobile genetic elements which can be found in almost all organisms. They are often referred to as jumping genes because they can move seemingly at random around a genome, jumping from one location to the next in the DNA. Their existence was proposed by Barbara McClintock, and in 1983, she received the Nobel Prize in Physiology or Medicine for this discovery. Transposons are found in almost all living organisms. In fact, remnants of transposons make up around 50% of the human genome sequence. So as you can imagine, the evolutionary pressure to silence active transposons in any cell is immense, as mobilization events can be highly mutagenic and deleterious. However, the flip side is that the ability to move around a genome might have some evolutionary advantage, mainly because they allow for genetic variation when mobilized. There are two distinct classes of transposable elements, which will be defined as Class 1 and Class 2 for this presentation. Class 1 transposons move by first being copied into an RNA intermediate through transcription, and then that RNA is copied back into the DNA through reverse transcription, using the enzyme reverse transcriptase. This reverse transcriptase is typically encoded by the transposon. After transcription is complete, this DNA is then integrated back into the genome. You can think of it like a copy and paste mechanism, just like you can copy and paste text with a computer. Now, I created an enlarged image of a Class 2 transposon, and it's shown at the bottom of this slide. You can see that this transposon contains a gene that encodes transposase. Flanking the gene at the 3' and the 5' ends of the transposon are terminal inverted repeats. These sequences are reversed complements of each other, which means that you can see a certain sequence at the 3' end of the transposon, and then you will see the complement of that sequence in reverse order at the 5' end of the same strand of the transposon. This inverted repeat serves as the transposase binding site. Transposase requires the presence of these specific sequences to cleanly cut the transposons out of its initial host site. You will also notice that the transposon is flanked by direct repeats, which are a result of the insertion of the transposon into the DNA. These direct repeats are not part of the transposon, and they are left behind when the transposon leaves this side of the DNA. This is much like a cut and paste mechanism that you can do with your computer, and in fact, Class 2 transposable elements are often referred to as cut and paste transposons. Transposable elements can be very difficult to detect in genomic data because they can be very repetitive in nature. Furthermore, the problem arises when these methods locate transposable elements in many different ways, which often lead to different transposable element detection rates on the same genomic sequence, and each method might output their data in a non-standard way. This makes it incredibly challenging to validate results pertaining to transposable element detection rates relative to other methods, and can also make further analysis of the data a bit trickier. There seems to be a trade-off between transposable element detection rate and transposable element positional accuracy within the genome, with compromised accuracy correlating to an increased number of false positive transposable element detections. Now this is where my research begins. I have been tasked with using the McClintock Pipeline, which was not made by Barbara McClintock, but rather named after her for her original efforts in transposition research. The primary goal of the McClintock Pipeline is to lower the barrier to installation, use, and evaluation of multiple whole genome shotgun resequencing database transposable element detection methods by automating the formatting of key input files and standardizing the output of these multiple methods to allow easy comparisons of results between them. The output file format chosen to standardize results for all component methods is a zero-based BED6 format because it allows easy integration with BED tools and the UCSC genome browser. All things said, the pipeline will automate the use of 6 different programs used for transposable element detection, and it will output a result that is in a user-friendly manner. From the paper accompanying the pipeline documentation, I have chosen Figure 5 for reproduction. It's a box plot representing 93 strains of yeast ran through the pipeline. The x-axis represents the programs and the y-axis represents the numbers of non-referenced transposable element insertions per strain, and this is predicted by the McClintock component methods in real yeast genomes. The authors of the research paper, who were also the creators of the pipeline, use 93 strains of Saccharomyces cerevisiae originally annotated in a different study by Strope et al. The EBI sequence read archive was given, but maybe more importantly, they included a script that would take care of running the entire pipeline for me, and I'll call that the main script. And a part of that script is dedicated to obtaining FASTQ files for each strain through file transfer protocol, so basically using wget from within a script, and this script, in theory, would also run the entire pipeline from start to finish. So, in other words, the script would obtain the data, which were paired in sequencing reads for each strain present in the original study, and then would use the reads as some of the parameters for pipeline execution. For starters, I used Argonne for all of this, which is our high-performance computing at the University of Iowa. To run this pipeline, I followed the instructions on the McClintock Pipeline GitHub page. This included me downloading Miniconda so I could use different environments for each of the 6 programs. The authors felt it was necessary to have virtual environments for each of the 6 programs to help with reproducibility. Having separate Conda environments for each method would allow me to download different versions of the same package or program and keep them separate from one another to minimize dependency errors. It also allowed me to have the same versions as was intended by each method at the time of their creation. Then I cloned the McClintock GitHub repository and simply ran the 2 included installer scripts. One was for the setup of the environments just described a second ago, and the other installed the pipeline itself. Before I could run the pipeline, I had to change the file paths in the main scripts to match my Argonne directories where things were either installed or located, such as my output directory where I would want the FASTQ files to be located, or even just the pipeline itself. This was all pretty straightforward as the paths were used as variables in bash scripts, so I just had to change the paths at that one place and that seemed to fix it. I finally got the pipeline to run. For starters, there were really actually 2 main scripts. I know I just talked about a main script, but there were really 2. They were both included for me to use, like I said. The first one obtained reference transposable element annotations and canonical transposable element sequences provided by yet another study, the Carr et al. Study, along with the author's manually produced hierarchy file based on the annotations from that same study. Then it made a text file with URLs on separate lines that would later be used to download each of the 186 FASTQ files, 2 for each of the 93 strains. This is the script that I actually ran to obtain my results, because at the end of that script, it called another script with parameters passed into it from that first script that were then used in the second script, including file paths and the URLs. The second script actually started by downloading the FASTQ files in a compressed form, then it uncompressed them. It then submitted jobs to the Argonne cluster scheduler in the form of a file path to the McClintock Pipeline executable with the reference genome, reference consensus sequence, reference transposable element locations, and the forward and reverse read for each strain as parameters. In other words, this second script submitted 93 different jobs to the scheduler, with the only difference in each submission being the yeast strain used. After each of the 93 pipeline runs were finished, I was left with 93 different directories, each one containing 6 files, 1 file for each program. The line count of a bed file, -1 because the top line is just a descriptor, will correspond directly to transposable element detections, each line counting for 1 transposable element detected. However, I finally figured out that I only wanted the line to count if it corresponded to non-reference transposable element insertions, because that is what the original file is built upon. To do this, I used this grep command shown on the slide, which iterated through all directories containing bed files, so for example ngstEmapper, and counted all instances of the word non-reference and appended that value to a file specific for just that program, with each strain represented by a line count on its own separate line. I did this for each program, each with separate files. I then sorted each separate file so that the smallest number was on top and combined the sorted files together with the result of having a 6-column CSV file with the corresponding method name at the top, with line counts being transposable element insertion counts sorted below. I didn't include the code in this presentation only because it's sort of complicated and it's not completely necessary to understand what I'm trying to do, and it is more of just ways to work around certain formatting issues and is also available on my GitHub if you're interested. Anyways, the example CSV on the slide only shows 11 lines, but in theory the CSV file would have 93 lines, not including the method names at the top. I then used various R commands to create my version of the original box plot. I am even using exact colors, which are represented in hexadecimal form, each color denoted with a hashtag in front of it. The original figure is located on the left of this slide, while the recreation is right next to it on the right. Because I used the exact same colors as the original, found using an online color picker tool, it is pretty straightforward to see that these two box plots are quite nearly identical. I have highlighted with the red boxes the differences between my recreation and the original. One difference is although the programs are in the same order along the x-axis, sub-labeling containing either group of split read or read pair are missing. I have added them in red to show that they would be similar in that regard, with a few possible tweaks in my R script that I just could not figure out. That's just formatting stuff correctly and more or less above my knowledge. The biggest difference is with the temp program. In the original, temp results were partitioned based on whether they had split read evidence or not. My recreation did not show any split read evidence. Considering I ran the program exactly as described, I'm not entirely sure if the pipeline was liable or if I just missed something critical when counting results or plotting with R. Just to be sure, I reran the pipeline many times with different parameters than what I was given for reproduction. And I even went so far as to just download the temp program itself and run temp only, and I couldn't seem to produce a similar result to the authors of this paper. The biggest struggle for me was R. Although I learned so much about R while trying to reproduce this image, I struggled for quite a while trying to figure out how to format my box plots. It also bothers me that they aren't more similar, especially surrounding temp not showing any split read evidence in my recreation. The authors did include an R script, however I couldn't get it to work for some reason pertaining to file paths and or file names. I tried moving bed files around as well as trying different working directories, but I couldn't get the script to work. That's why I analyzed the pipeline output the way that I did. It was probably a super challenging way to do it, and it's also probably not perfect, but it's all I could do at the time. And if temp has something to do with the pipeline run not working, then the authors really need to update their scripts for running the pipeline properly. Otherwise, everything else was very well documented, and outside of having to change file paths in the pipeline scripts, everything ran like it was supposed to. If I were to do this again, or if I had a little bit more time to devote to this, I would want to go back to the beginning. I would probably just delete everything and install the pipeline from scratch, because I truly do want to know what happened to the split read temp results, and something just isn't right there, and I'm wondering if the McClintock program just did not install correctly. Also, the requirements for programs used within this pipeline were sort of strict. If they didn't have great documentation or widely available code, being that of free or open source, they were out of the running to be included. Further, if they required any wet laboratory enrichment, they were also disqualified. And many of the rest of the programs were thrown out because they were too difficult to install. The list of the potential programs went from 33 to 6 real quick, the 6 being the only ones that met each criteria mentioned. I would be interested in doing this in the future because there is potential for new methods or programs to be added to the list, and it would be compelling to see how those additions would impact the results. All in all, it was a very worthwhile experience, and outside of the temp results, I feel as though the authors truly put a ton of effort into this rapidly growing opportunity. I appreciate you watching my video and thank you for your time.